Hello, this is class Bio 225, Genetics from Genes to Genomes, Chapter 7, Part 5 with Dr. Lucy Abgaryan. Until now, we've extensively talked about different mutations, mechanisms of mutations, and all about gene sequences and different defects of it. Now we will talk about some early techniques for detecting and determining where the mutations are. Those are early stage techniques. One of them is rapid mapping method. Please note that this is a very crude method for understanding where approximately the mutation is located. It's not a very accurate method for determination the particular mutation. And also it's important to know that this is not what current science can do. The current sequencing techniques are so much more developed. This is just part of history of genetic science. So remember, for DNA duplication process, one of the DNA strands is always the wild type, the parental one, and due to replication process, if there is a mutation with deletion, we will regain the wild type, full wild type again. So by doing random mutations, introducing random mutations, when we have the mutation in the strand where the deletion was, then that will give us a possibility to still have the, due to recombination, we still have the strand that has the wild type, and it will give us the ability to regain the wild type DNA. In our model organism, which was the T4 phages, due to this technique was discovered that there are fragments of sequences that are more inclined to have point mutations than others, and these fragments of sequences are called hotspots. We think that mutations are bad. However, mutations gave us also a lot of insights into science of genetics. For example, some diseases are a result of inborn errors of metabolism. So, in which case gametes themselves have the mutations that zygotes get from them, and so the progenies are having the mutations now. So it was discovered that the one gene, one enzyme concept. So it is known, we know from previous classes, that metabolic processes are heavily relying on as enzymatic reactions. So if there is one enzyme that is dysfunctional, and it can be due to particular mutations, then entire system is collapsing. Entire metabolic process will be dysfunctional. So the hypothesis, and we talked about this particular case previously, the Neurospora crassa, where the mutants in arginine synthesis occur. So by doing genetic dissection of biochemical pathway, we can determine in which particular enzyme the mutation exists that is causing the dysfunction of the system. So just like saying very schematically, so like there are, let's say, there are multiple enzymes involved in the entire metabolic process, particular metabolic process, and the chain and that process is a chain reaction. So let's say there is enzyme 1 that requires pathway that requires substrate 1. So we do the substrate reaction of substrate enzyme reaction, and if function goes through, there is a product appearance, which is becoming substrate for enzyme 2, and so for by looking at this entire pathway with enzyme 1 to enzyme N, we can see where the disruption is happening, and that's where our problematic sequence is. That's how the mapping is done in order to determine the mutations in which particular enzyme the mutation is. In some cases, mutations in gene can result in amino acid substitutions that disrupt the function of the encoded protein. And the sickle cell anemia is caused by such disruption. It's caused by missense substitution in hemoglobin beta, and the missense substitution is substitution of amino acids where one amino acid is substituted by another amino acid that has physically it has absolutely different physical properties. For example, hydrophobic amino acid is substituted by hydrophilic amino acid, or acidic amino acid is substituted by more basal amino acid. Here is the schematic representation of the enzymatic chain reaction that I was talking about, where there is a chain reaction, and for entire process different enzymes are involved, and if everything is normal and there are no mutations, then it should go from the very beginning, from enzyme 1 all the way to the bottom and get to the CO plus water outcome. However, if there is a mutation, which is the case for alkaptonuria, there will be there is a mutation, and in halfway through the whole pathway, and that will cause in malfunctioning of the entire process. Now let's look and see how Biddle and Tatum came up with the hypothesis one gene, one enzyme. They used, as a model organism, they used Neurospora, and they looked at it under X-ray influence, and X-ray radiation causes disrupted synthesis of arginine in these organisms, and that by itself is resulting in generating of two different strains. One is prototrope, which is the wild-type strain that grows in minimal media without additional supplements for growth, and another strain is auxotroph, which is the mutated strand that cannot grow in minimum media. It requires some additional supplements for growth. And in order to understand the mutations, they used the recombination analysis, and they mapped mutations and were able to recognize that there are 4 different regions of genome that are responsible for this event, and the 4 different regions were arginine E, arginine F, arginine G, and arginine H. So here is a question for you. What is the mechanism that is explaining this? To be more clear, here is the formulation for the question. So we've seen that there are 4 different regions of genome, ARG, ARG-E, ARG-F, ARG-G, and ARG-H, that are involved in synthesis of arginine for Neurospora organisms. So mutation in any of these genes, any of these 4 genes, will result in dysfunction for arginine synthesis. Question is, why all 4 genes are needed to be complete and not mutated in order to have proper arginine synthesis. And here how the experiment was conducted. So there was the wild type Neurospora that was exposed to X-ray radiation, and it's now we have mutagenized conidia. As we know, Neurospora is a fungus, and then it was crossed with it was sexually crossed by opposite wild type, and the fruits were collected, and then they were transferred to tubes of complete medium, and they were inoculated with single ascospores. And then after complete media, they were transferred to minimum media that didn't contain the necessary growth nutrition factors for growth of the fungus that is missing arginine. However, they didn't know that at that point, and then they took samples of that culture, and they added to each one of them, they added different amino acids, and they saw that by adding arginine they can see the regrowth of the culture, which indicated that the arginine synthesis of arginine was affected by X-ray induced mutation. So coming back to our example where we had 4 different genes for arginine synthesis, so now we can understand which biochemically active molecules or pre-products for arginine are result of which particular gene. So if we grow with nothing in it, we see basically no growth of our strain. When we add ornithine, that's when we see growth of we still don't see any growth. And then the same with citrulline or arginine succinate. However, when we add arginine succinate, we see that we are having growth of arginine E, arginine F, arginine G, but not arginine H. That's how we determine that that's the product for that's what arginine H gene is responsible for. It's responsible for synthesizing arginine from arginine succinate. And then we can add citrulline in it, and we can see that the citrulline is responsible for citrulline to arginine succinate transfer. Arginine G is responsible, gene arginine G, and so forth. The same with ornithine, arginine F gene, and for so when we have a mutation of arginine E, arginine F, arginine G, and arginine H, that's why in any of the interruption in any of these genes will result in interruption of the arginine synthesis in general. And then in the bottom schematics, you can see that more clearly that how the enzymatic reaction goes. Arginine E will cause the acetomethane to synthesize ornithine, and then arginine F will result in synthesis of citrulline from ornithine, and then arginine G will be the enzymatic transfer to arginosuccinate, and then arginine H gene is the enzyme that is responsible for synthesis of final product arginine. I know that we have talked about amino acids during our first class. However, I want to go over this again briefly, just summarizing it. So we know that there are 20 essential different amino acids, and each amino acid is containing three groups of molecules. It's the amino group, the N terminus, it's the carboxyl group, the C terminus, and also it's the variable R group, which is the different side chains that are unique for each amino acid. So again, we have talked about this. The amino acids are distributed in different subgroups depending on their physical properties, and it is determined by their R groups, by their chain groups, and there are non-polar amino acids based on that. There is class of amino acids that is uncharged, natural amino acids. There is a group that is charged, basics and acidic ones that are positively or negatively charged. And when we are dealing with mutations, depending on what type of mutations we are dealing with, it can be very significant or it can have very minimum impact. So let's say if we are having a change of amino acid within the same class, let's say a basic amino acid lysine is being substituted by histidine, for example, then the impact can be not as significant as if charged amino acid is being substituted by uncharged amino acid, for example. So again, sickle cell anemia, a great example for amino acid substitution mutation. So this is disorder where the red blood cells are having dysfunction in transporting oxygen, and this takes place when the glutamic acid is substituted by valine. And the thing is that the glutamic acid is an acidic amino acid, which is charged amino acid, and it's substituted by valine, which is a non-polar amino acid. And as you can imagine, that can cause a significant change, modification in a 3D structure of the protein, which by itself is causing disability of binding the oxygen molecules to the beta chain of the hemoglobin. Therefore, the transport of oxygen is being disruptive. Sickle cell anemia is pleiotropic. If you can recall, we have talked about pleiotropism, and the pleiotropic is when mutation in one gene can cause dysfunction in other proteins, other functions that seem to be unrelated to the mutated gene. So therefore, sickle cell anemia, being pleiotropic, is causing different dysfunctions within the organism. And here is the map presented with different dysfunctions that are due to sickle cell anemia. So in order to understand why is that, we have to remember how the proteins are structured. So first of all, we know that there are interactions in polypeptides within the amino acids, and that is the primary structure of the polypeptides. And then besides that, there is a secondary structure, the alpha helices, the beta pleated sheets, and we have talked about all of this during our first class. And then after the secondary structure, there is the tertiary structure, which is basically the complete 3D dimensional arrangement of the polypeptide, where you can see like how a single peptide is structured within the 3D dimensions. However, in order to be a functional protein or functional system, peptides are not, mostly they are within the groups, so different peptides will come, or different peptides, different proteins, or subunits of the same protein will have to come together and basically form this quarterly structure, which is the final functional stage of the protein or the system that is going to be functioning. And this is how it is in our organism, the final stage of functioning proteins. Here is another example for polytropy. So let's look at interleukin receptor 4, interleukin receptor 2, and interleukin receptor 7. And those are the cytokines, interleukin 4, interleukin 2, and interleukin 7, that are interacting with these receptors. Those cytokines are basically involved in cell cycling processes very intensively. If we look at the structure of the receptor and in a binding, the binding packet for interleukin, we can see that the receptors are pretty different from each other. However, there is one subunit that is exactly the same throughout these three different interleukin receptors. That's the gamma subunit. So therefore, if there is a mutation that is causing a dysfunction in gamma subunit, it's going to affect binding and functionality of all three receptors that we are discussing here. So seemingly unrelated mutation is going to result in dysfunction in seemingly independent from it events, and that is how you can imagine the polytropy, where the mutation in a secondary structure of gene sequence is affecting due to involvement in four-dimensional final functional structure of the protein of the system. Okay, now let's summarize what we have talked about today. One gene, one enzyme. It's a concept that is not broad enough. So first of all, not all proteins are enzymes. And also some proteins are multimeric and subunits are encoded by different genes. As we have seen already, complex pathways can be dissected through genetic analysis. Also, we have seen that different mutations in single gene can produce different phenotypes. Examples like case of interleukin Ch07 or the sickle cell anemia. Different amino acid substitutions, such as basic to basic or basic to nonpolar or positive to negative, all of these different substitutions can have a very different broad spectrum of effects on protein function. Mutations can affect protein function by altering the amount of normal protein that is being produced, that is being made. And example of it is in case of cancer, where let's say the growth factors are affected, and there is uncontrolled amount of production of growth factor enzymes, and that is causing the growth of tumor or something that is uncontrolled.